I would like to welcome everyone to our fourth webinar. Today we'll have Dr. Bruce Smith from the College of Veterinary Medicine presenting the Auburn University Initiative in Cancer. The webinar will last approximately 1 hour. All webinar participants are initially on mute, and you need to unmute yourself during the presentation if you have a question by clicking on the green microphone button on your dashboard. There will be a question and answer session. We'll start at the end of the presentation. You can also submit questions for Dr. Smith in the questions section of the dashboard, and they'll be read by Ms. Vicki Kretschmeyer, our webinar moderator. The webinar will be recorded, so it can be viewed later on the Auburn University Research Advisory Board website. You're encouraged to pass the link to this webinar and the previous webinars we've had as well to any colleagues that you know that may be interested in the subject matter topic. And if you know of any people or organizations that may have interest in the research topics discussed today, we'd ask if you'd please get that information either to Dr. Smith, to myself, this is Rodney Robertson, or to Vicki Kretschmeyer. And we'll send you a short survey at the end of the webinar, and we ask if you would please complete the survey so that we can get the feedback and make sure that we improve the webinars in the future. Today's speaker is Dr. Bruce Smith from the College of Veterinary Medicine. Dr. Smith is director of the Auburn University Research Initiative in Cancer. Dr. Smith graduated from Harvard College with a bachelor in biology. He received his veterinary medicine and PhD in genetics degrees from the University of Pennsylvania. Dr. Smith joined Auburn University in 1993, and he's currently serving as a scientist in the Scott Ritchie Research Center and the professor of pathobiology at the College of Veterinary Medicine. Dr. Smith is also the director of the Auburn University Research Initiative in Cancer. So Dr. Smith, we'll turn the webinar over to you at this time.
All right, that's good. Can everybody hear me?
Yes.
Yes, I can hear you.
Yeah.
Great, thank you, Rodney. Let's just bring up the first slide here. All right, I'd like to thank you very much for this opportunity to talk to you a little bit about what the Auburn University Research Initiative in Cancer, or ORIC, has been doing, especially some of the successes that we've had in the past year. I've spoken with you folks before for at least a brief time when you were out here at the college this spring. And so what I want to do with this presentation is really bring you up to date on some of the more broader issues with this program. So I'm going to divide this talk into three parts. The first part will be a little bit of history about who we are and where we came from. The second part, I want to talk a little bit about some of the measures, the metrics of excellence that I think we're just starting to record now because we're a brand new program, but we'll hopefully over the long term give you an idea of our success and certainly give us an idea of what we're doing right and what we're doing wrong. And I hope you'll be pleasantly surprised by the initial metrics. I certainly was. And then finally, and perhaps most interestingly to all of you, I'd like to share with you some of the really interesting science that's going on as part of the program. And so probably the last half or so of this webinar will be a review of about 8 different investigators' work and just giving you a broad idea of the kinds of projects that they're working on. So with that, let's get started on this webinar. So most of you are familiar now probably with the new Auburn University marketing program called This is Auburn. And so I wanted to use that as an opportunity to kind of introduce you to our program and where we're going with it. So I'm going to switch over and play a quick YouTube video here, if all the technology works the way it's supposed to, which I think it will. Most of you have probably seen this video. I hope you have. If you've been to a football game or watched a football game, you have.
This is my first step toward a fulfilling and successful career.
This is our one medicine approach, which links discoveries in one species to advance in health in all of the species.
So hopefully you recognized somebody on that that brief shot, even if it wasn't me, but I was in there in the middle, right in front of Cam. But what I wanted to say is that that commercial really does hit home the point of ORIC, which is the idea that we're talking about a One Health program, and so this is One Health and this is ORIC, and our goal is to talk about a One Health approach to cancer and cancer therapy. What is One Health? It's really the combination of veterinary medicine and human medicine into a single field. The concept is pretty clear, and that is that what we learn in human health we can apply to veterinary medicine, but what we learn in veterinary medicine we can also apply to human health. And so it's a synergism. It's a field where we're able to work with veterinarians, with pharmacists, with nurses, with physicians, all working on the problem of cancer. And it doesn't really matter if it's cancer in a person or cancer in a dog. Those two organisms in particular, but any organism gives us more information about cancer and more information about ways in which we can fight cancer across the breadth of species. So we have from the very beginning had this One Health, One Medicine concept for ORIC, and we strongly believe that's a core of our program. And one of the strengths of the program here at Auburn is that we can bring this breadth of knowledge and breadth of interest across both human and veterinary medicine to the problem of cancer. So just want to mention a little bit about how that works. We talk a lot about animal model species, and out there in the science world, a small animal is a mouse or a rat, a rodent model. So that's down here at the bottom of the picture. And typically, treatments these days for humans often get tested in mice or in rats and then transferred directly to the human patient. That has a success rate of less than 1%. So 99% plus of the treatments that are tested in mice never make it in humans. And the question has always been, how can we improve that? And one way to do that is to look at an intermediate species, like the dog. And we say intermediate because in my world as a veterinarian, the dog is a small animal, and in the NIH, NSF, extramural funding world, the dog is considered a large animal. And of course, here at the College of Veterinary Medicine, a large animal would be a horse or a cow, and that's not quite where we're at. So we've coined this term, intermediate animal model, and that really refers to dogs and cats. And they're intermediate for two reasons. One, the dog is intermediate in size between the mouse and the human, so it gives us a scale-up potential. We can look at what dose we used in the mouse and decide what kind of an increase in dose do we need to use to get to a dog, to get to a human. And also, it's intermediate in terms of the developmental pathway, in terms of the translation of something from the laboratory into clinical use in humans. We can use the dog as a stepping stone to better understand the way in which these treatments work, and as a consequence have a greater chance of success when we do try to translate that to humans in the clinic. So we really like to think of Auburn as operating in all three of these areas. We have basic science, we look at rodent models, we have the capability through the College of Veterinary Medicine to do translational intermediate models in the dog, and finally we are interested in translating all the way to human patients. And in fact, I have this bidirectional arrow where the emphasis is not just on translating from the dog to the human, but also from the human back to the dog, so that both species can benefit. So let's talk a little bit about ORIC. The program was officially founded January 1st, 2012, so we're not quite done our second year at this point. The reason for this program coming into being was primarily because Speaker of the House, Mike Hubbard, was involved in the budgeting process and recognized that cancer research was occurring at Auburn and looked for a way to help fund that. And so Mike was a driving force in getting the original funding and subsequent funding for us for this program. We found out about that funding in mid to late 2011. Dean Calvin Johnson of the College of Veterinary Medicine asked me to head a panel that put together a strategic plan. We touched base with various stakeholder groups, put that strategic plan out, had it reviewed, and were able to return a plan back to the dean within about 2.5 months. So pretty remarkable, actually, for a faculty committee being able to put together a strategic plan in about 2 months. I was very proud of the committee. At that point, Dr. Johnson created the program. We recognized immediately the program was not a College of Veterinary Medicine program, but should be an Auburn University-wide program. Cancer is certainly much, much bigger than the college, and so before we ever even started working on this program, we invited people from across the university. Again, in that first year, we received $1 million in an appropriation from the state of Alabama. This year, that was increased to $1.1 million in the current fiscal year, and that was additional money was care of a collaboration between Speaker Hubbard and Senator Tom Whatley. And Senator Whatley was looking for ways to also invest in the magnetic resonance imaging facility, the MRI facility on campus. And so $100,000 of that funding was specifically earmarked for the MRI facility, and I'll talk a little bit more about that later. And then in this coming fiscal year, we've been awarded an additional appropriation of $1.1 million. So total Alabama appropriation to the ORIC program is now at $3.2 million. So significant state funding to help us move cancer research forward at Auburn. And again, we owe a lot to Speaker Hubbard, Senator Whatley, and the Auburn University governmental relations folks, namely Sherry Fulford, who shepherded this through and was really the main contact. So all of those people really deserve a tremendous amount of credit for this. Moving on, I think when I last reported to you, I said something like ORIC had over 60 members or over 65. I'm happy to report that as of this week, we are over 85 members, and we continue to grow. So that's very encouraging. That's a fairly good segment of the population here. Over 50 of those members are, in fact, faculty. The rest are staff, students, postdoctoral fellows, a broad range. And I was quite pleased and surprised when we initiated the program to have staff members come up to me and say, well, I know this is really a faculty research thing, but cancer is personal to me, and I want to be involved, and how can I be involved, and can I help you? Can I do administrative things for you? And it's just a great part of the spirit of ORIC and the people who are involved to have people who are, secretaries, receptionists, typists, come up to me and say, I want to be part of this program. This is interesting. This is neat. What can I do to help? And we've really tried to foster and encourage that. We currently have 6 colleges or schools involved in ORIC. Love to have more. We're working on that. Those 6 include the College of Education, namely the Department of Kinesiology. The Samuel Ginn College of Engineering has been involved from the very beginning. The College of Human Sciences, Harrison School of Pharmacy, again, involved from the beginning. College of Science and Mathematics, or COSAM, as many of you know it. And finally, the College of Veterinary Medicine, where, again, we were involved from the very beginning. In addition to Auburn University, we now have members at 3 other universities spread across the United States, from near to far, from UAB to St. Louis, and that is also an opportunity that's growing to build new collaborations outside of Auburn as well as inside of Auburn. That's clearly a direction we want to go in. With ORIC, and I say we a lot when I talk about ORIC, because while I'm the director, we also have a board that helps me with the program. That consists of 4 faculty members who are cancer researchers, and an additional 4 ex officio members who represent College of Pharmacy, College of Engineering, the Associate Dean's Office here at the College of Veterinary Medicine, and then Dr. Robert Judd, who is the director of the Boschel Diabetes and Metabolic Disease Program. And he's involved because we think that metabolic disease and diabetes have a very, very close link with cancer, and also he's got a successful program, and so he's been able to help us and advise us in ways in making our program more successful. And I think you'll see later when I talk about some of the science how that metabolic disease aspect has played out and in fact is prominent in what we do in terms of our research. With that group and the overall group, we took what we want to call a deliberate, logical, and thoughtful approach to funding cancer research. That is, we did not go out and say, okay, let's give each of the investigators involved X amount of dollars, but instead we looked at ways of managing our funds and using our funds in a very appropriate way, that where we did our due diligence ahead of time, where we knew that putting money in these places would be likely to be generating extramural funding. So some of the criteria that we use are leveraging of funding. So one of the things that I'm always thinking about is how can I make my buck go further? Who else's money can I use? And there are several ways we've done that. And of course, what that does is it takes our money and it spreads it out, and it makes it have a bigger impact if we can pair with people. And there are some great examples I'll talk about a little bit. One would be the intramural grants program here at Auburn that's run through the Office of the Vice President. Another would be those MRI funds that you see in our budget for this year and the next year. The other thing that we look for are sustainability plans, and that's not sustainability really in the buzzword of green that's being used now, but you could almost think of it that way, but rather program sustainability. That is, how can we translate the funds that we're providing to investigators here at Auburn into something that is a continuing source of funding for us from extramural sources. And primarily what we're looking for is federal extramural dollars from major granting agencies or from large national and international nonprofits. And what we really want to do is see a multiplier on our money. So if whatever we invest in an Auburn investigator, we'd really like to see that come back several-fold over that in terms of what their external funding is. And of course, you all recognize that that brings in things like indirect cost dollars, salary savings dollars, that all help to build the research enterprise here at Auburn. So that's a big component. We're also always looking for interdisciplinary approaches. These days in science, if it doesn't show some nature of interdisciplinary approach, you're pretty much dead in the water with the extramural funding sources. So we do really emphasize that, and we've really tried very hard to bring groups together and had great success at that. Really very, very pleased with the way in which that has come about. And with that, of course, comes the collaborative nature of science, where we do get people together and start talking about science and put new ideas on the table. That's really the way in which new technologies and new intellectual properties are created. So it's really important to us that we create a collaborative environment. And again, I'll mention a little bit more about that later, because I think we've been successful, at least in the beginning. We need to build on our success, but we've had some great success in doing that early on. And finally, we wanted to make sure, I talked about this, doing our due diligence for funding. We want to be sure that our review process is very rigorous, that we're preparing investigators for extramural reviews. It's not appropriate in my mind, and certainly in the board's mind, to simply give out money because we have money and people need money. We really do want to focus on providing funds for good science that's been reviewed, that we think has a good chance of being successful. And I want to emphasize that that's not necessarily low risk. One of the problems that you always fall into when you want to be assured of return on your funding is to go with the low risk stuff. But in fact, some of the high risk stuff can pay off even more. And so we want to be very careful to be rigorous, but not too conservative. And then I note constructive in our review process, and that's about growing the faculty here. We want to be able to have people understand what they need to do to improve their grants, their proposals, in order to get funded. So many times when we have a grant program that's run through ORIC, we will have several rounds of submission and review prior to an award. And that's happened now in most of our programs, where we will get proposals in, they will go to a review panel, they will be reviewed, and then sent back for corrections and improvements to the investigator, re-reviewed, and only when the reviewers are satisfied that changes have been made that will make that proposal a better, stronger proposal is the funding awarded. So we really are trying to take a very rigorous but constructive approach to doing reviews. This year, we started reviewing some metrics for the program. Now, remember that we were started in January of 2012, so in January of 2013 was the end of our first year. And because we were a new program, we didn't really have the opportunity to develop metrics for the program before it was started. So we wanted to look at metrics for the first year. We sent out a survey to all of our faculty members that included quite a few questions about their research and research productivity. Excuse me. Out of the approximately 40 faculty at that time who were eligible to participate, we had 15 return answers. So a little over a third, and for those of you who send out surveys like Rodney will after this webinar, a 1/3 response rate is actually pretty encouraging. Typically response rates hover around there or even lower. And we were very pleased to see that of those 15 faculty, they had submitted 46 extramural grants, and of those 46, 19 were funded. And that's a 41% funding rate. That is significantly higher than the national average. And what that tells me, at least about those 15 investigators, is that they're pretty good at what they do already. And hopefully we're going to build upon that to create more successful investigators. The real surprise to me was when we looked at total dollars in extramural funding. And for the 2012 year, that was $3.53 million, or a little over 3.5 times the investment of $1 million that the state had put into our program. And understand, this is our first year, so that investment really hasn't even had a chance to work very much in terms of time. And so that's really kind of a baseline point. We hope that our extramural funding goes up from there. I think there's every indication that it will, but we'll have to wait and see. We also wanted to ask about relationships. Remember, I indicated collaboration is very important to us. And so we were able to identify the fact that there were 31 new internal collaborations. Now, I want to caution you on interpreting that a little bit because, of course, if you and I are both investigators at Auburn and we start a collaboration and we both report, we're both going to report a new collaboration. So that 31 collaborations may indicate somewhere less than that, however many I don't know. And certainly, again, only 15 out of the 40 eligible reported, so there may be collaborations that were formed that weren't reported. But still, that's a pretty significant number. And that means we are talking to each other and we are getting collaborations started internally, which is very exciting. On top of that, we had 7 new external academic collaborations and 5 new external industrial collaborations. And those are just new collaborations for the 2012 year. So that means that our people in OREC are not just talking to each other, but we are also going outside of Auburn, and outside of Auburn to universities, and also outside of Auburn to industry. And I think that's a very important combination. A measure of our scientific productivity, our scholarship, is the number of peer-reviewed papers. And for 2012, that was 53. And I remind you, that's for 15 individuals reporting. So that's well over 3 papers per person per year, peer-reviewed academic publications. So that's pretty good productivity on average. And in addition, another surprising number for me were the number of national and international presentations, and that was at 49 for those 15. So again, more than 3 presentations per person out of those 15. And that means that our faculty are being recognized and asked to speak at national and international meetings. So that means OREC has a national and international audience for our research, and we're being seen as part of those presentations. Those individuals are being encouraged to use our logo and our name and credit OREC if they have OREC funding, obviously. So OREC's image is also being seen in that scientific venue, and it's building a knowledge that cancer research is happening here at Auburn. I think that's a very strong advertisement for cancer research and for Auburn University and research in general at Auburn. Training is an important component of what we've been thinking about for this program and how we're going to move forward. We look at it as training the next generation of scientists who will work on cancer. But of course, the other part of training is that these are students who are working in the laboratory right now, working on cancer problems for faculty members of ORIC, and as a consequence, they're conducting research and they're doing productive things. And again, we had a little surprise here because the largest single group of students involved are undergraduates with 38. We then have also 8 master's degree students being mentored and 27 doctoral degree students being mentored, as well as 8 postdocs. So that also gives you a pretty good idea of the breadth of the academic mentoring that's going on within the program and the amount of student involvement. I'll mention when we get to the grants some things we've been doing to get doctoral students involved, but clearly with that number of undergraduates we need to think about ways in which we can more officially involve undergraduates and serve their needs. And so in fact I'm working with Dr. Ray Henry right now to get us more involved in some of the undergraduate research programs because I think an official interaction is appropriate when we're working with that many undergraduate students in our research laboratories. So I want to move on to the research grants program that we've been running and tell you about all the different ways in which we're taking the funding that the state has given to us and trying to make that work in a research environment. And so rather than running a single grants program, we've run several programs now, and I hope you'll see that the intent really is to both go after the big dollars in a forceful and yet appropriate way, but at the same time also try to grow the program, to create new investigators, to bring people into cancer research, and also to take some of our folks who've been involved in cancer but not very successfully at the federal level and really work with them to try and improve their research programs and their grantsmanship in a way that will enable them to go out and get federal funding. So the first grant program that we offered was what we called the IGP Match. And the IGP stands for the Intramural Grants Program. That's the program run by the Office of the Vice President for Research. I'm sure most of you are familiar with that program. At most levels of funding, that requires some sort of match from the college, school, department, etc. of the investigators. That's also an interdisciplinary inter-college program in most cases, and so it requires part of that match come from multiple different colleges or departments. What we did as a program was offer ORIC members 50% of the match that was required by the Vice President's office. We asked that they submit a letter of intent and that letter of intent detail precisely what they were going to be doing and submitting for the intramural grants program. Those were reviewed and 4 of those were then offered matching funds. And so we committed a total of $143,500 to 4 different proposals that were then submitted to the intramural grants program. I was disappointed, but only one of those was funded. That was a $50,000 match from ORIC, and that led to the purchase of an ultra-high pressure liquid chromatography triple quadrupole mass spectrometry system. I will tell you that I know that that's a really neat machine, and that it's being used, or it's being installed actually right now, and is being listed and will be used by many, many different investigators, not just in cancer, but across the breadth of the university. But in the ORIC program, there are quite a few investigators who are looking forward very anxiously to the installation and opening of this facility, because it is a major piece of equipment, a major, major improvement on what we've had at Auburn. That is being installed in, I believe, Rusty Arnold's lab in the College of, in the School of Pharmacy. We have just issued our call for 2013 because the Vice President has just issued his call for the intramural grants program. So we will be doing much the same in the coming year, hopefully funding additional projects from ORIC members and helping out with the match. to me, this is a great way of leveraging our money. We're putting in a small portion of the total grant, as I say, usually about 25% of the total, but the benefit is that we're going in and hopefully helping our investigators write better grants by pre-reviewing them and also by providing them with some of the matching funds. Talking about supporting graduate students, I mentioned that earlier. We created the Speaker Mike Hubbard Fellowship in Cancer Research to recognize Speaker Hubbard's efforts on our behalf. I will emphasize that because we were concerned about conflict of interest, what we've done is create a separate funding source for that fellowship. It is not funded directly out of ORIC funds, but rather it is funded out of an endowment that folks can contribute to, and we are working very hard to build that endowment as a separate endowment from ORIC funding. In the meantime, the Colleges of Veterinary Medicine and the School of Pharmacy have contributed the funding to actually pay the majority of that fellowship. And this year, that was awarded to a graduate student, PhD student by the name of Farouk Kabir. Mr. Kabir is a graduate student in Dr. Kurt Bird's laboratory at the College of Veterinary Medicine and works on the genetic basis of breast cancer. And I will actually show you some of his data later in this presentation. In addition, we have decided that funding a graduate student was such a great idea and such a good investment of funding monies that we have announced the ORIC graduate fellowships. These will be up to an additional 4 graduate fellowships restricted to PhD students. We have just issued the request for proposals. The deadline is the end of this month. And we will be selecting, as I say, up to 4 fellows. And we always like to phrase, you'll see me phrase our grant requests that way very frequently, or our grant definitions that way. And that's part of being rigorous about our reviews. If there aren't 4 qualified fellows, we will not fund 4 fellows just to fund 4 fellows. We will fund as many qualified fellows up to 4 as we identify. And that funding includes full salary support for 1 year and is renewable for up to 3 years with sufficient adequate progress and a positive review from the panel and from the mentor of that student. So this is one way we hope to contribute both to graduate education but also to help ORIC investigators get a graduate student in their laboratory, because that can often be one of the most difficult things to fund, is to find funding to support it. On top of that, that is the beginning of a graduate program for ORIC, which could lead to the ability for ORIC to apply for federal funding for training grants, something that we don't really have right now and something that we could very much use. Another set of grants that we have awarded already are grants for magnetic resonance imaging in rodent models of cancer. And this is where that $100,000 that Senator Whatley got added to our budget comes into play. I spoke with Tom Denny of the Magnetic Resonance, the MRI Research Center, and basically offered to leverage his money and my money. And we offered $100,000 of ORIC money in addition to that $100,000 that was appropriated for him if he would put his money into a MRI of Cancer grant, and he thought that was a great idea. So we combined forces and contributed $200,000 out of that $1.1 million and identified 2 projects. Each received $100,000. One entitled Magnetic Resonance and Bioluminescent Imaging of Mouse Models of Primary Cancer and Metastatic Disease that's run by Dr. Peter Panizzi. And the second, quantification of magnetic nanoparticles in rodent tumors with MRI by Dr. Alan David. Dr. David is in chemical engineering. Dr. Panizzi, I believe, is in the School of Pharmacy. And so both of these projects are now ongoing. The funds have been awarded, and you'll actually see something, a little diagram from Dr. David in a little bit about what he's doing. Then we wanted to find a way to get smaller amounts of money to larger numbers of people. We called this our seed grant program, and that's because we're planting a seed and hopefully growing a big idea. The idea here is, of course, that these are a little bit less mature kinds of ideas or proposals. They may be higher risk, but they also may be higher reward. We're still looking for these to be translated towards extramural funding, but obviously $20,000 isn't necessarily going to be enough to walk out the door, get some data, and then go find an NIH R01 or an NSF large grant at $1 million or more. So this might be the kind of funding that then leads to a foundation grant of $100,000 or $200,000, and then ultimately, hopefully, to an NIH or NSF grant in the $1 million+ range. We were able to review quite a number of grants and selected 11 that were suitable for funding. Those 11 grants came from 9 different investigators, so 2 investigators did get funding for 2 different proposals, and we did make sure that those proposals were in fact different, that they were not overlapping, and those projects are up to about $20,000 each. So with those 11 grants, we invested about $220,000 in what we called seed funding, or the idea funding, trying to get a new concept out there. No need for preliminary data, but hopefully at the end of the year, there is a requirement that these grants would have a requirement, rather, that they have a plan for extramural funding, and hopefully at the end of the year, some of these will be mature enough to go out and seek extramural funding. And so we're hoping next April to be looking at the results of this and probably offering this type of grant again, maybe more than once a year, to try and get an active rotation going with lots of these small grants going. And this is really our attempt to also grow the program, to put some money out there to get new people involved, people who've never done cancer research. There's no need for you to be a superstar in cancer research to get one of these grants. You simply have to have a great idea, even a good idea, an interesting idea, and it has to be cancer. And so somebody who's never done cancer before, hopefully we're going to bring them into the program through this type of a grant program. We also have a collaborative research project grant program through the Southern Research Institute. That came about actually through an interaction with Jack Sechrist of SRI through the Research Advisory Board. So this is one of your success stories. And Jack put me in touch with Mark Sudo at SRI, and Mark and I have been working on this for almost 2 years now. And again, this is an example of our constructive grants process. They're looking for new ways to use their libraries of chemical compounds. They have about 1 million compounds in various libraries that could be tested for their efficacy against cancer, for example. And what they're looking for are new assays, and what we have are creative people who have systems that might be used for assays, and our idea was to pair the two. And so we solicited up to 2 proposals of $100,000 each. In fact, we have 2 proposals that have been through an iterative review process where they've been reviewed several times in collaboration with SRI, and those now are pretty much in their final status, and they're ready for resubmission and re-review, and hopefully in the next few weeks we will complete that process. As part of this, we're putting in $100,000 to each of the investigators, and Southern Research has agreed to provide the screening portion of this as their cost in kind and not charge the investigators for the screening. And of course, intellectual property results will be shared. Southern Research is very agreeable and an easy group to work with respect to intellectual property. Really pleased with the way in which they handle it and the way in which they handle collaborative research. Great bunch of people. So hopefully next time I speak with you, we'll be telling you about the success of those projects as well. Finally, I'd like to tell you about what we're calling our Major Grants Program. This was in response to a challenge from a couple of different individuals that said, go big or go home. And so this is our Go Big or Go Home project. The idea here is that nobody is more successful than people who are already successful. That is, people with a track record who know what they're doing, who've been through the federal grants process, who sit on federal boards, federal review panels, or other panels, and as a consequence, they end up knowing how to submit grants and how to make these things work. So what we're looking for here is grant proposals of up to $100,000 a year for 2 years. The PI must have a track record. These proposals must be designed to lead directly to significant extramural funding, and by significant I mean in the $1 million+ range. We have plans to fund up to 3 in the current cycle, And right now we have 5 proposals that have been submitted after a pre-proposal cycle. And so we have what we were calling an NIH-style review panel pending. We just assigned reviewers this week, and hopefully we will be having a panel meeting in about 3 weeks and be able to select the 3 best of those 5 proposals and move them forward. One of the things that was really encouraging about this was that we had a pre-proposal requirement and a pre-proposal meeting requirement. That is, the very first thing that had to happen was investigators had to come to a meeting. And I will be honest and say that I worried that probably 4 or 5 people would show up. And we asked anyone interested to show up, anyone interested in being a collaborator, anyone with some technology that might be useful. We had over 30 people. The meeting lasted almost 2 hours, and several new collaborations came out. It was really a very impressive group. We had each person present some of their ideas, and it's the kind of sharing, of collaboration that we're going to try to find ways to encourage on a more frequent basis. As I said, we have members from 6 different colleges, and at least 5 of those colleges were represented at that meeting. So it was an outstanding interdisciplinary collaborative meeting. And so hopefully we will, sometime around October 1, be awarding about $600,000 in funding. So this is real money, and this hopefully will lead directly to substantial extramural funding, as well as our other projects. All right, so I'm going to move on to talk about some science. I'm going to need to do this fairly quickly because I see it's already 2:40, but I want to tell you what we're doing because it's exciting. The first person I want to tell you about is Celisa Westrick. Dr. Westrick is at the School of Pharmacy, and she's interested in the delivery of service. This is not a basic science proposal, but really it's a patient interaction proposal. And what her research is looking at right now is the potential for increasing the rate of human papillomavirus vaccination. That's HPV. I'm sure many of you are aware that the vaccines were recently licensed, first for use in girls, but now also for use in boys as a preventative for cervical cancer. And it looks like HPV vaccination could be a very, very good tool in preventing cervical cancer. And so she's looking at the possibility of providing vaccines like this in a pharmacy setting, like we already do for influenza. So very interesting kind of a patient delivery side. And I wanted to bring that one out first because I wanted to show you that ORIC is more than just laboratory research or new drugs. We're looking at all kinds of aspects of cancer, including that patient delivery aspect. The next project I want to talk to you about comes from Dr. Elizabeth Lipke of Chemical Engineering, and she's looking at ways in which you can control or mimic cellular microenvironments. We grow tissue culture cells on flat plates right now, and that's really doesn't mimic a tumor at all when you have a flat layer of cells. And so what she's trying to do is create 3D tissue culture systems where you mimic the architecture of a tumor better, and then you can look at questions of delivery. So on this slide, we're showing some of the gels that she uses to do that. Up here next to the quarter or some samples, and you can see that you can control the size of these gels very easily. They're easy to manufacture. And over here on the lower left, we've got some panels that show breast cancer cells growing as colonies, as round groups, and colon cancer cells growing the same way in these 3-dimensional engineered tumors. And so they have very interesting and uniform properties. And if we look at the next slide, they've looked at a whole bunch of parameters, and I don't need to go through them, but basically they can identify the concentration of oxygen, for instance, the accumulation of lactate, a result of metabolism, and a lot of other parameters that really indicate that these things look like tumors. And if you look at this graphic that's moving down here in the lower right corner, you're going from inside one of these to outside. So the red dots on the inside are cells that are dying. They're called necrotic cells, and that's very typical of a tumor. To the outside, where these cells are multiplying quickly, that's the zone of rapid growth. Again, very, very classic of tumor growth. So these new 3D models of tumor are extremely hot research tool. They're just coming out now, and people are very interested in them. So it's wonderful that we have Dr. Lipke here at Auburn, and she's collaborating with a number of investigators using this system. Next, I want to mention Rusty Arnold. I mentioned him a little bit earlier in some of the equipment. Dr. Arnold is working on a way to enhance delivery of drug. We have a number of different ways in which people are doing this, and Dr. Arnold's approach is to wrap drugs in a number of different lipids. And so if you look over on the left of this figure, the X's are the drug, and they're all wrapped up in these little circles, which are different kinds of lipids. The red one is a particularly important one because it's targeted by an enzyme that's on the cell, and that's a phospholipid targeted lipid. And then these big green blobs are also part of the targeting system. And in these tissues where there's high vascular permeability, namely tumor, these complexes float around in the blood. They can come out of the blood and into the tissue. They then bind to the cell through these little Pac-Mans. Once that happens, these red circles are degraded by the enzymes that are more common in tumors than they are in normal tissue. That releases the drug out into the area around the cells, and that leads to more delivery of drug to the tumor, and also because of this binding, an internalization of drug into tumor cells. So there's two different ways of killing tumor here. We're going to increase the concentration of drug around the tumor and also deliver it directly to tumor. This means that you can target tumor, but over here in normal tissue, we have very few of these complexes getting through because the vasculature is not leaky, and also we don't have the targets here on the surface of the cell for them to bind to. So we're going to limit drug uptake in normal tissue and spare that normal tissue. Dr. David, who I also mentioned, is in the chemical engineering department, is interested in magnetic nanoparticles. He's created particles which have an iron core and then have a coating of a variety of different substances. One of the things he's doing is also looking at targeting tumor, but here he wants to image tumor. And he's created a particle over on the left which has this iron core, and that's visible through MRI, and a fluorescent imaging agent that's stuck to it through a linker. And if you put that into a slow-growing tumor tissue, a number of the magnetic particles go in, but the tumor does not mobilize these fluorescent molecules, and so you don't see very much fluorescence. In an intermediate grade tumor, you're going to see more fluorescence in the tumor because it starts to degrade these nanoparticles and remove the fluorescent molecules and allow them to start fluorescing. Then in a high-grade, high-proliferation tumor, you'll see a lot of the fluorescent particles because they've been removed from the nanoparticles, but the nanoparticles are still there. And so what he's measuring is the ratio of fluorescence to MRI visible nanoparticles. And as the fluorescence goes up, it means the tumor is more aggressive. And so this might be a way of, in vivo, in live individuals, identifying the aggressiveness of a tumor before you ever have to take it out surgically. Dr. Allen's also looking at ways of targeting various drugs to tumor. One of the issues here is we treat a whole human. It's going to take 6 grams of drug. If I just treat the tumor itself, it only take 100 milligrams. If I just treat the cell, it takes 2 milligrams. And if I'm at the subcellular level of treating parts of the cell, it might take a fraction of that. And so what he's doing, working with Valerie Petrenko, is identifying a bacteriophage in a library that basically contain protein sequences and looking for the sequences that either bind to intranuclear targets or, intranuclear, intracellular targets and cellular targets. And he's identifying those phage and then trying to link them to his nanoparticles. Dr. Mansour at the College of Veterinary Medicine in Anatomy, Physiology, and Pharmacology is looking at prostate cancer, and he's identified a very interesting phenomenon, which is a plant lipid, stearidonic acid, or SDA, when given to prostate cancer cells, these are normal prostate cancer cells in green, does slightly reduce the ability of those cells to grow. Doxorubicin in blue is an anti-cancer drug, and that does a better job of killing those prostate cancer cells, excuse me. But if you mix the two together, SDA and doxorubicin does a fantastic job, even better than either of the two by themselves. So it's a synergy of killing those prostate cancer cells. And he's looking for answers as to why that works. And what he's found is that they work together to activate a nuclear receptor called PPAR gamma, and you can see the PPAR gamma levels in this slide by the bright red. And so down here in the left corner where you have SDA and DOX, you get the most bright red. That means the most activation of this gene, and that gene is involved in cell death. So he's starting down the biochemical pathway of understanding how this synergistic treatment for cancer might work. Dr. Mike Green of Nutrition and Food Science in the College of Human Sciences has created a fatty nude rat model. And the nude part is not because we're, well, we're not into titillation or anything like that. Nude animals, nude mice, nude rats lack a thymus, and as a consequence, basically lack a functional immune system. What that does is it allows us to inject cells that are of a different species, and they will continue to grow. So it allows us to use those animals as models. And here you can see his fatty nude rat is quite a bit fatter than the normal wild type. What he's doing is then looking at tumor growth in these animals. And here we're using the IVIS Illumina in vivo imaging system that's been partly purchased through ORIC funds to image tumor cells inside a live mouse. So the red is tumor cells. And you can see from day 13 to day 25 how that tumor in that one animal grows. And we can monitor that over time. What we see, here's going to take away your appetite for what you ate for lunch. If we feed these rats a high-fat Western diet or a low-fat Western diet, we can watch tumor growth. And what Mike has seen is that in the high-fat Western diet, tumors grow faster, that's the red line, compared to the low-fat Western diet. And if you take the tumors out at the end of the experiment, the small tumors are from the low-fat Western diet, and the large tumors are from the high-fat Western diet. So as of today, I've sworn off cheeseburgers for lunch. A couple more to show you here, and we'll go to some questions. This is some experiments by Dr. Bird. I mentioned that Mr. Kabir had contributed some of these. This is his work, and they're looking at gene expression in cancer cells. And so bottom line is he's looking at, in this panel of little red lines, he's looking at products of genes or the presence of genes in canine mammary tumors. And there's several different lines that we're looking at, 9, 12, 27, 28. These are all just different tumor lines. And NCF is normal dog fibroblasts. And basically what he's showing is the places where there aren't lines, those genes are missing. They've been mutated, they've been inactivated, and that's part of the cancer pathway in these dog tumors. So he's identifying the steps that have made these tumors into cancer genetically. At the bottom, Mr. Kabir has been looking at something called microRNA, or MERS. This is a very new, very hot area. These are not RNAs that lead to protein production, but they're regulatory RNAs, and we're just beginning to figure out how they work. And they're looking at a panel of 277 known microRNAs and looking at the level in tumor cells compared to normal cells. And so where you see green dots, it means the level of this MER is elevated in a tumor cell. And where you see red dots, it means the level of that MER is decreased. And so this is just a preliminary screen that's giving them a great idea of where to go to look at changes in regulation of tumor cells. Dr. Bird has also created what he calls the barking mouse. And that is because he's taken a nude mouse, one of those mice that does not have an immune system of its own, and he's implanted into it a canine immune system. So in these graphs in the middle right, what you can see is in the right-hand graph, the cells that are boxed, this is a blood sample from a mouse, those are canine immune cells in that mouse. So he's created a mouse with a canine immune system. He can then transplant a canine tumor into that mouse and ask questions about how that canine immune system responds to that canine tumor. A very important difference than asking how maybe a mouse immune system would respond. And so it's a really interesting way of looking at it. And again, this is our in vivo imaging system. It's allowing you to see this green tumor blob on the chest of this mouse. Dr. Bird's also been working on a vaccine for breast cancer, and hopefully he'll be reporting those results very shortly. But this is in a clinical trial using patient dogs, client-owned patient dogs, and actually vaccinating them against breast cancer. Finally, I want to mention Dr. David Reese. Dr. Reese is working in a protein called ERB-B4. The ERB family of proteins is well known for its causative role in cancer. And so in colorectal cancer, EGFR or ERB-B1 is known to be a causative gene. In breast cancer, ERB-B2 is known to be causative, and in melanoma, ERB-B4 is thought to play a role. Now, there are drugs right now that block ERB-B1. That's called Erbitux or cetuximab. There's a drug or an antibody that blocks ERB-B2, Herceptin. That's maybe a drug that you've heard of. But there are no drugs that block ERB-B4. And so what he's looking at is ways to block ERB-B4 and perhaps use those as a treatment for melanoma. Oops, I forgot one other research program, my own. We currently have a clinical trial going in dogs with osteosarcoma with a conditionally replicative adenovirus. That is a virus that is a live replication-competent virus, but it only replicates in tumor cells. And we give this to dogs that have been diagnosed with osteosarcoma after they've had their primary tumor removed. And if we look in patient blood after we give the virus, we're looking for the amount of virus in the blood, and we see early on there's a little tiny bit in the blood, but at 24 and 36 hours there's a peak of virus in the blood. That falls off very rapidly at 48 hours. But by 72 hours we see another peak of virus in the blood. And what this is this is virus that's now replicating in tumor cells in the dog. And when it does that, it kills the tumor cells. It bursts them like a balloon. And we also think it involves some immune system responses. So hopefully we're creating an immune response to the tumor as well. And we'll skip that slide. And then I just want to sum up by saying that I think the road ahead is really bright. I think we've established our program in the last year and a half, year and 3/4. It's very exciting work. We keep growing, and I don't really see major obstacles to what we're doing. And we continue to look for collaborations. We look for collaborations in a variety of different places, industrial, academic, and we're just extraordinarily excited about the kinds of things we're doing here. And with that, I'm happy to take any questions.
Are there any questions for Dr. Smith?
Right in the hour.
Okay. Well, Dr.
Wow,
Smith,
we're all asleep.
okay, we certainly appreciate it. Not everybody has the ability to ask questions, or they can't actually send the questions to the moderator, but some people do not have microphones, and sometimes with telephones we have trouble getting questions in. But
Rodney, I would also say that if any of you do have questions, ideas, suggestions, interests, please feel free to contact me directly. It's easy enough to find me. It's Bruce F. Smith. There is another Bruce Smith on the faculty, and I'm at the College of Veterinary Medicine. The email for me is smith, S-M-I-T-H, B as in Bruce, F as in Frank, at auburn.edu.
Well, Dr. Smith, we certainly appreciate your time today. This webinar has been recorded, and it'll be available on the the OVPR Research Advisory Board website, so other people who were not able to attend this webinar will have the opportunity to go back and view it in the future. Our next webinar will be October the 9th at 2 o'clock. Dr. Dave Beverly will present his work on GPS and vehicle dynamics research. Dr. Beverly is recognized as a GPS expert, so we'll get information out, the link out. I think the link's already out right now to register for the webinar, but as we get closer to the time, we'll get information out about to make sure that people are aware of the time of the webinar and have the information they need to register for it. Thank you very much, Dr. Smith, and thank all of our participants.